Excellent. So I will talk a little bit about how we utilize NGS here at Novo Nordisk, and I've selected a subtopic of that, and that's around protein engineering and design. It's a little bit different from the previous speaker, of course, but please interrupt along the way and let me know if there are any questions as well. So just feel free to keep this very interactive. I just want to give a short and brief background to Novo Nordisk. We have been around for almost 100 years now. So it started when insulin was discovered in Toronto and was brought back to Scandinavia by August Krogh and Marie Krogh, who brought that back. And clearly our focus is to use technologies to make the life better for our patients in the metabolic and rare disease space. So just for everybody to understand what kind of company we are in the metabolic disease area. So the corporate strategy is to make medicines that improve the lives of our patients. That means that we have a very strong focus around diabetes care, obesity care, and also within biopharm, where we focus on rare diseases and are also looking into other rare complications as well. Last but not least, just to augment a little bit on this, we look into these serious chronic diseases. So we are addressing unmet needs within cardiovascular disease, non-alcoholic steatohepatitis, NASH, Alzheimer's, and chronic kidney disease. And our pipeline is around these things, but also one thing that we have, and that's what I'll come into now, is why are we interested in NGS and applying NGS to the different pipeline. So where do we use NGS in our drug discovery and design pipeline? So there are like three areas where it's completely critical for us to have access to this technology and where it's really empowered us to do new things that we're not able to do previously. So clearly in the early target discovery, we use NGS quite routinely now with transcriptomic data in order to figure out what are the different disease associations, how can we get a better and deeper understanding of the biology involved here. And then also methylation profiling in order, again, to get a better idea about different targets, identifying new targets, understanding the biology. So that's one part where we really routinely use NGS and the results of that. And that enables us to discover new targets, new associations, even new causal things between our genes in order to come up with new targets that can improve the lives of patients. Then clearly, once these have been identified, the next big thing is, can we then start to design drugs for that? And Novo Nordisk is a biologic company. That means that we are mainly focused around peptides and proteins. So in order to do that, we have different display platforms where we can then engage with these targets. We have phage display, yeast display, mRNA display that we run routinely in order to actually generate new targets towards these or new drugs towards these targets. But once you get a potential drug out of this, it needs to be optimized. So that's the third arm where we really use NGS quite a lot. That's in order to optimize our drugs using, again, display platform, but using the NGS technology in order to actually fish out and improve our drugs there. So I'll go a little bit into that here in this talk. The way that we set this up here at Novo Nordisk, I think my slides, are they not being...
No, we're still on the driving change slide.
Okay.
There we go.
Okay.
We're corporate strategy and disease areas.
Yes, okay. I see there is some delay. Can you move them forward, Alfred, or how's that possible?
I don't have control of that. Which slide do you want to be on at this point?
Slide 6.
Could you tell me? Okay, thank you.
Or if you just call out a new slide, then we can advance.
Yeah, okay, so slide 6, if we could go to that one, and then I'll say new slide. I do apologize for that. I don't know why that's not working here. Yeah, and then just go through it, just step through it until we are at the end of this one. So what we do is that we've implemented this design, build, test, learn cycle at the Novo Nordisk. And in order to do that efficiently, we try to use computational as much as possible and then combine that with high throughput experiments. If you press next, that would be good. So what we do initially is we start with a structural interaction of the desired target that we have identified through early target discovery. As you can see here, you have the main interactions between the target and then the hotspot residues, which are the one we either identify through computations or from existing crystal structures. So that's our initial hypothesis generation. If you go to the next, press next. Then the next part of this is that we start to do in silico design of these proteins and peptides. So what we do here is we use a computer to actually figure out how can we have scaffolds or peptides that can then support these interactions in order to bind the target of interest from us. So when we do this, also because computational algorithms are still a little bit noisy and unprecise, we need to test quite a lot of designs. So we can order these designs. I'll come into detail how we actually go about this, but this is a step where we actually do NGS on that. So we do an experimental testing of these. Normally that can range between 10,000 to 100,000 in this case. And then we use, as you can see here, then we use fact sorting. In one case, we can use face display or mRNA display. Doesn't really matter, but then we use fact sorting in this case in order to actually identify what binds and what kind of binders do we have in this pool of design. So then after identifying that, we use, again, different computational methods to actually start to mutate the whole protein and peptide in order to figure out where is it possible to actually improve these molecules. What you see here on the bottom, you see a heat map on different positions that have been identified, where green are improvement and red are positions that are not able to be touched for a particular molecule. Then we take this data that's been through the NGS, and then we use this data to actually guide us to a better molecule. And one of the things that, of course, we look for as these molecules have to go into patients is, are there any T-cell epitopes? Are there any production issues? Are there any chemical or biological liabilities, et cetera, et cetera? And then we use the NGS data to actually guide us to remove these and improve the molecule also with respect to potency and efficacy. And this is all done in a data-driven feedback to improve the molecular design. So we have actually merged NGS with machine learning in order to actually optimize these molecules. If you go to the next slide. Yeah, so I'll go through two case studies here where we will show how we actually go about this. So case study one is de novo design of compounds. And what do we mean when we say de novo design of compounds? That means that these are compounds that are not existing in nature. So these are something that we create in silico in order to have the right efficacy and potency and desired properties for our patients in order to actually improve the life with whatever disease they're living with. So one of the ways that we do this is to do computational design, because the space and the complexity of this is so vast that we need algorithms to actually ensure that we can actually control this. So what we do is that we use these de novo design, and these can both be protein and peptides. In the previous slides, I laid out how we actually go about this in these design, build, test, learn cycles. We can also sometimes, and that's also an often problem, repurpose either existing things that could be, for example, different proteases that we are interested in. It could be antibodies that have been raised on experimental platforms, and then we use algorithms again to come up with hypotheses on this. So the next part that we do is we do library construction and size, and that clearly is something that is constrained by the different display platforms that we have. So we look into if we need to do degenerate codons, where we can do libraries where different positions are then varied. We can do oligo pools. That was the one I showed in the previous example, where we then order small oligos for 10 to 100,000 in order to look into these. And then last but not least, we can also do doped oligos where we control the library size much more refined. And that really depends on the problem. The question then becomes, how do we then read these things out? And that's where the NGS again comes into the picture. And how we utilize that is that we need to fish them out of these display platforms. And clearly there's a design decision here. Can we get away with short reads, or do we need to do long reads? And that clearly depends on the problem that we're trying to solve. Again, for the smaller proteins and peptides, which is something that Novo Nordisk focuses quite a lot on, we tend to get away with short reads in many cases. In other cases, if we're working on bigger proteins, that could be enzymes, for example, we need to do the long reads in order to be able to capture all the variation on this. Remember that what we are looking for in the third box here that I was explaining is variant differences between the different designs that we have. That could be enzymes and antibodies. So that's the problem, and how we start to design our initial workflow around this. So we take the size into account, and we take the sequencing technology into account in order to make sure that we utilize this in the best way. So if you go to the next slide. So here's an example from a yeast display. So here what you see is on the left, you see a real-world data where we do a FACS screen on this. So the first library is then screened, in this case, around 100 nanomole of the bait. And what we can then do is that we can put different evolutionary pressures on that. So then the pools here are then identified, and then we start to do deep sequencing on those in order to figure out which are the different sequences, how good are they, and what kind of enrichment ratios can we then calculate on these. And the reason for that is that we can then identify the most fit sequences under those circumstances that we are interested in. And that can be different things. So we can stress in the yeast display, we can stress our cell type under different conditions that we find relevant and that can translate into better molecules, but also just simply look at the more and stronger binding could be something, or weaker binding, depending again on the problem we're trying to solve. Again, the point is that we use NGS here to actually fish out and figure out how the different energies of these variants is. And on the right, you can see one of the examples where we can then do a analysis based on the NGS data to figure out which sequences are enriched in this space, and then go for, in this case, the most enriched sequences. If we go to the next slide. So the case study 2, so I just tried to present to you something where we are trying to do a de novo identification of a drug to a particular target. So in this case study here, what we use the NGS and these display platforms, again coupled strongly with computational algorithms, is to do optimization of hits. So the problem is that when we fish these things out from our initial display, they have the initial properties that we're looking for, but they are not finalized as a drug yet. So in order to do that, we need to do another round where we then look into how can we then optimize these things into becoming drug-like molecules that can then be tested in much better assays that are translatable for the different diseases that we are interested in curing. So the first thing that we do again is, because the space is so vast, even with the new technologies like display and NGS, we still need to limit ourselves with respect to the sequence space. So the first thing that we do is that on the right, what you see is a molecule that binds a ligand of interest. This ligand of interest have, of course, something that we deem first shell residues. Those are residues that directly involved with that. And we know from binding optimization that these residues are probably where it is most important to hit, for example, for binding affinity. There are also something in second and third shell, but just for case of illustration, what I show here is the first shell interactions. So we use computational tools to screen for those and figure out which ones are the most important and which other entities should we put into that. So that gives us a number of different positions and a number of different amino acid types that we need to substitute into this molecule in order to improve, in this case, affinity. That gives us a number of variants, and then we can, from this number of variants, we can then back calculate and figure out what kind of library construction should we actually do, what kind of codon should we do, as I mentioned in the beginning. If you go to the next slide. Here is then, shown the results from these deep sequencing and analysis of optimization of these different compounds. What you see here on the heat map is that on the top you have the different amino acid substitutions, and then on the rows you have the different positions. And the different colors then illustrate if something is depleting or enriching at these positions, meaning that it improves for the different things. So when we do the NGS analysis, again, we try to do a ratio analysis to figure out if different positions are improving or depleting this molecule with respect to the properties that we have. Once we have identified that, we can then start to re-optimize that. So that's a very easy way to get the first initial hypothesis generated around the molecule and how it should be optimized. Once we've done that, we then do a focus library. So just to give you an illustration about how strong this technology is in order to actually optimize the properties of our molecules. You see the SSM on the lower right. You see that the signal is relatively weak, and you figure something out, and then you do different rounds of enrichment here. But once you start to do the focus libraries, where you combine all of these different residues, what you clearly see on the left on the figure down there is that the binders start to be extremely potent during the FACS screen. And that's the one that we are interested in. So using this technology, we are then able to actually improve our molecules relatively very, very fast, and then come out with molecules that then become very, very drug-like, that we can then move on in our value chain. So if you go to the next slide. So what I'm trying to get at here, and what I'm trying to summarize at this last figure here, is how are we now going about this, because clearly new technologies are coming out there, and what we've seen is that the strength is in combining technologies between NGS and then especially computational algorithms for design of compounds. So initially, when we did this, and this was before the revolution of machine learning, which is something that has really also empowered next generation sequencing, especially in our field of biologics and optimization of biologics, we now have a pipeline where we use machine learning and physics-driven design and optimization on the molecules. Once we have that, clearly, as it was the scientific breakthrough of 2021, we use AlphaFold, which has really changed the confidence with which we can actually start to do these predictions. So there we use AlphaFold in order to guide us on how these libraries should be constructed. And again, we do that both on the monomer and on the complex, as you can see here. Why do we do it on the monomer and the complex? Clearly, we need to do it on the monomer as well, because that's the stable state of the molecule, and you still need something that's stable in formulation. So we need to understand how these molecules react on that. And then we combine these into computational design. So we add in data from different sources, et cetera, and that gives us the library. So as I showed in the two case examples, is that this is extremely important for us to then throw that through a display system, get some learnings from that, do NGS, and then use the NGS analysis to actually show us by using statistical and machine learning algorithms, how can we then re-optimize these molecules in a faster and smarter way. Also, given that this generates so much data, it's something that can then be transferred project to project, meaning that we start to have more generalized learnings about proteins and peptides in particular. So that's the pipeline that we have, and where it's clearly using both the strength of machine learning, computational tools, and of next-gen sequencing. If we go to the next slide. Yeah, just want to conclude here. What we have seen, especially with NGS and how it's a vital part of everything that we do, is that we can then accelerate the generation of biologics through this computational design. And as I mentioned earlier on, computational design is still very noisy, and that means that we need to look at big numbers, and that can only be done through NGS. So it's a vital part of these pipelines that I've just shown you. Deep sequencing enables this fast evaluation of millions of sequences, which is necessary also for us to start to build learnings around proteins and peptides and how they behave both as a single structures but also in complexes. So that's clearly extremely valuable for us to have access to these data. And then last but not least, we are doing this is the why we're doing this, is to engineer smart and innovative molecules that can actually benefit patients in our disease areas. So there are, at the end of this value chain, actually people that needs this and that benefits from these technologies, which is extremely good, because we can then start to target our molecules to be much more specific and be much more regulated with respect to their properties and what they can do. So that is what I would like to show here. If you go to the next slide. I want to thank you all. We are hiring, and if you have any questions, please contact me or one of my colleagues. Also, if you're interested in jobs at Novo Nordisk, then please reach out also again to me or my colleague, and please visit our website if you have any questions regarding us as a company. So last but not least, thank you all, and if there are any questions, please just ask them or reach out to me on my email after this presentation.
Thank you so much. That's amazing. As we wait to see if there are any questions that pop up, I'm put in mind of the famous Thomas Edison quote, where he said, "I've learned 2,000 ways not to make a light bulb." I think he would be fascinated with this technology about how it can accelerate that. You know, instead of groping around in the dark with your eyes closed, you know, using these incredible tools to narrow the search and get to your target so much more effectively.
I think that's a great effort. I made comment on that. I think it's a great analogy because it is really two technologies that converge and supplement each other so well. And it's if the darkness and Thomas Edison, it's like all of a sudden you've been running around the room with a small light or flashlight, and all of a sudden you just turn on the light, and then you can actually start to see how the room is and how things are connected. So I actually like that analogy very much, and that's exactly what it is. It is technologies converging such that we can actually ask better questions and make, you know, in this case, better drug for our patients.
Excellent, very exciting. Looking forward to seeing what this new approach can yield for everyone. Well, no questions show up, so we're going to move ahead.